Assalamualaikum warahmatullahi wabarakatuh and good day to our panel and friends. Today I will present a review on the photocatalysis inhibition of E. coli by using titanium dioxide photocatalyst. My name is Mariam Amira binti Abdurrahim. I am from AS222 degree of Bachelor Science on Chemistry with Management. My supervisor name is Dr. Wan Izhan Nawawi Wan Ismail. Pharmaceutical industry develop and produce market drugs for use as medication. However, it leads to the contamination of water, where bacteria will affect the organism as the consumer consists of human and livestock. E. coli is a most common bacteria found in human or animal feces. Several wastewater treatment techniques that can be used, which are chemical technique, biological technique, and physical technique. In this research, we will focus on the chemical technique. Chemical technique consists of photocatalysis, ozonation, photon pattern, ion exchange, and adsorption. We will focus on the photocatalysis in this review. What is photocatalysis? Photocatalysis is a process to initiate a reaction in the presence of UV light to oxidize the organic material into carbon dioxide and water. How does the photocatalysis work? When we excite the photon to the semiconductor photocatalyst, electron from the valence band will excite to the conduction band. The electron will react with oxygen to produce hydroxyl radical, while hole will react with water to produce hydroxyl radical. These radicals are very strong and enough to reduce the organic material into carbon dioxide and water. The TiO2 semiconductor is the famous photocatalyst that be used due to its low cost, abundant, high stability, high photocatalytic activity, and non-toxicity. However, it has its own limitation where the wide bandgap energy makes high energy of photo-generated electrons. This makes the TiO2 photocatalyst only function under high energy of UV light irradiation. Next, recombination of electron hole. Increasing in recombination electron holes makes the photocatalyst less active along photocatalysis process as more electron going down from conduction band to valence band. We want the electron at conduction band still stay at the conduction band to produce the hydroxyl radical. Due to the problem statement, we focus on the TiO2 modification by using metal and non-metal dope, which are silver dope, copper dope, cerium dope, neodymium dope, and nitrogen dope. All these modified TiO2 can decrease the bandgap energy and reduce the recombination electron holes. The target of this modification are as preparation method for photocatalyst inhibition of E. coli. The best modified TiO2 under photo inhibition of E. coli. The optimum condition of E. coli photo inhibition. Research objective is to identify different methods in preparation of modified TiO2 for inhibition of E. coli by using metal and non-metal dope TiO2. Second, to compare the best method of preparing modified TiO2 under photoinhibition of E. coli. The third, to understand the behavior of modified TiO2 by using SRD and photocatalytic activity as support data for the optimum condition of E. coli photoinhibition. Analysis and interpretation. All these graphs show the optimum condition for the photoinhibition of E. coli. The first graph is for the silver dope. The second one is for the copper dope in terms of ofloxacin OFS. The third graph for the cerium dope and the neodymium dope. The last one is nitrogen dope TiO2 NDT. The table shows the result from the graph. The silver dope TiO2 completely degraded at 10 minutes. The copper dope TiO2, the zone of inhibition is 18 mm at 180 minutes. The cerium dope TiO2, the inactivation rate of E. coli is 1.302 log CFU per liter at concentration 0.5 gram per liter at 180 minutes. The neodymium ND dope concentration at 2.0 millimolar show the zone of inhibition 28 mm, while the nitrogen dope TiO2 inactivation rate is 8.5 exponent 5 CFU at concentration 0.2 gram per liter at 10 minutes. How the TiO2 degrade the E. coli? This diagram shows the E. coli structure consists of DNA, ribosome, cytoplasm, plasma membrane, capsule. This diagram shows the general degradation mechanism where TiO2-based photocatalysts surround the cell membrane and absorption on surface occur, and then reactive oxygen species generated will attack the cell membrane, lead to destruction of cell membrane, and then enter the inside of cell, lead to deactivation of cell. To more understand the degradation mechanism, this diagram shows the cell wall of E. coli consists of outer membrane, peptidoglycan, and plasma membrane. At the outer membrane, lipid peroxidation occur, where the free radical OH steal electron from lipid in cell membrane, resulting in rupture of cell membrane. The product is lipid peroxide. This diagram shows the reason why silver dope TiO2 have been chosen as the best modified TiO2, as the silver ion inhibit the cell at 3 parts, which are oxidative stress caused by the reactive oxygen species OH radical. Second, interaction Ag+ with the thiol group in proteins. And the third, destruction of bacterial cells via strong affinity interaction between Ag+ and cell membrane. Ag+ ion also inhibit the DNA ribosome that lead to the destruction of cells. In conclusion, modification TiO2 by metal and non-metal dope TiO2 have shown their ability to deal with bacteria in variety ways, especially by using light to generate the reactive oxygen species able to kill the bacteria. Among the TiO2 modification by metal and non-metal dope, silver dope TiO2 have been proven to be the best modified TiO2 with the highest degradation of E. coli, as it takes only 10 minutes to completely degrade the bacteria. The modified TiO2 nanoparticle on the surface of the material can also serve as electron traps, improve the separation of the electron holes, and decrease the recombination rate. This helps to improve the inactivation rate in comparison with undoped TiO2. That's all from me. Thank you.